Hey, how are you? My
Hey.
name is Daniel.
All right, nice to meet you, Daniel.
Nice to meet you. How are you doing?
I'm good. How are you?
Fine.
That's good.
So today I want to tell you about DNA replication
Okay.
and what's replication.
Replication is just like duplication.
So just as you stated, I'm gonna talk about how DNA. Every cell has the DNA inside the nucleus. So for it to go from generation to generation, when the cell divides, it has to replicate its own DNA so that the genetic information can be carried out from one cell to another or from the parent to the offspring. So how does the cell do this? We know that DNA it's a double strand, like it has some bases which are basically some molecular structure, and it occurs at it's twisted around each other.
Mm-hmm.
So for it to be copied, it has to be unwind. Like for the replication process to happen, it has to unwind the twist. Like imagine like a zip of a bag. It's always closed, but in order to open the bag, you have to open it. So this DNA has to be opened for it to be copied. So
Mm-hmm.
it's now double stranded, like this. It has shown the structure. But when it's open, it becomes single stranded.
Wow.
So it can be copied only when it's single-stranded. So
Okay.
how do we go from double-stranded to single-stranded? We have to, there are certain proteins which go into this DNA and attach to it.
Mm-hmm.
And actually this protein is a complex of other 4 proteins. Those different proteins are within that complex structure have different function. One protein will unwind this one, will change this one from double strand to single strand. So that process is called helicase. Basically, it helps unwind from double strand to single strand. Remember, before even this one starts, it has another protein. This complex has to come and attach to the double strand. The first event that occurs, it binds to a certain region of the DNA. How does it recognize this region? There's a certain sequence in that DNA that is recognized for it to bind.
Mm-hmm.
Yeah. So once it's bound to the DNA, it starts opening up this from double strand to single strand, which is done by helicase. Okay, now it's open. What's the next step? We have to copy this information from each strand. So the way it does this, it lays down a primer. Primer is basically, it's like a sequence of a DNA but a short strand, like a building block. So when it lays down this, it starts forming fragments that joins into this. So this protein that initially bound to the DNA, it's just going ahead while adding new strands, like making new strands. So from this one strand, it's gonna make a new one, and from this other one, it's gonna make a new one, right?
Mm-hmm.
So, but one thing I didn't mention, when it goes from double-stranded to single-stranded, this is more unstable. So it has to be stabilized by other proteins, which are also within this core protein. So they are called single-stranded stabilizing proteins, binding proteins. They stabilize this DNA. Okay, so we opened our zip and we are trying to copy this. But one thing happens is that when these single strands are being copied, one will be synthesized. Imagine it's like this. It's going into this direction. So there's a certain way DNA is copied in a 5' to 3' direction. But this will be 3' to 5' direction. So if it's being copied, it will go from 3 to 5', and this one will go from this one. This down will go from 5' to 3', and we need to generate a 3' to 5' direction, right? Like a certain direction had to be maintained. So what's gonna happen on the other strand is gonna, instead of laying a primer here and going this direction, synthesizing a new DNA in this direction, it's gonna lay it in front and synthesizing backward. So again, when it reaches here, it lays another one and goes into back direction. So what will happen is one strand will be like it's going a long sequence without breaks in between, but the other one will have like interruptions, you see. So we can't have DNA like that incomplete. It's incompletely made when this one is completely synthesized. So there's another protein that's gonna help in that. It's gonna come and replace these strands with to something that is similar like this. Like overall, it's gonna find a way of changing it to a certain way it looks similar. So okay, replication goes ahead until it reaches a certain point. Remember, when you started replicating, it recognizes a certain sequence. That's for it to start. It couldn't start any position. It has to start at a certain place. The way that means it also has to stop at a certain position. So when it reaches this position, it notices another sequence that, let's say this is like a red stop sign that says stop, and that's where it stopped replicating. So this we started with double strand. It was open to 2 single strands, and then each strand was copied. So if we make a 1 copy out of each strand, how many will you get? This is okay. This is 1 copy, 1 copy. So I'm gonna make a copy of this and a copy of this. So we went from 2 to 4, right?
Mm-hmm.
So this like the parent strand that uses the best to copy. So these 2 will be joined together to form a double strand. So we have our new DNA here made from the old one.
Right.
Yes, and it uses energy from ATP because all this process take energy.
Okay.
So
Wow.
do you have any questions to ask?
So you saying all this so it depends. Our DNA depends on our parents' DNA by
Right.
this structure.
Yeah.
That's what you're saying.
So it's gonna copy almost the same sequence in it. So whenever it detects like an error, like it doesn't copy exactly the same, there's another enzyme called DNA polymerase. It comes and corrects those errors once it's made.
Right.
Yeah. So the parental DNA, not necessarily the parental, like the old one is used to make a new one.
Mm-hmm.
This one again will be used to make a new one.
Right.
So in that process, it's gonna be replicating every time.
Okay. Okay. Wow, this was a lot of in-depth information because I never really thought much of it, but I see, and you understand it very well, which is good.
Did I make any sense at all?
Yeah, it does. It does because at first I was like, "What is he talking about?" But then when you took your time and you explained it, and now I understand, and I also see how you joined 2 together and it made it, a replication of it, which was good. I understand.
Okay.
Any other questions for me?
Okay, I'll ask you. Okay. Don't worry, Zach, I'll edit that part.
Okay. Hey, look, we just cut it off.
Wait,